Hi everyone, this is going to be a talk about cellular injury, so the different ways that cells can become damaged. And before there is damage to a cell, the cell will change to adapt, and we're going to call those things adaptive changes. So for your minimum learning objectives here, we'll talk about how different agents of injury can lead to damage to the cell or adaptation to the cell. And it's important for you to recognize an adaptive tissue response, meaning the body is trying to get used to having some sort of irritant in its vicinity and keep the host safe, and what constitutes an irreversible injury, which would hopefully lead to cell death. So in pathology, we need to know what these things look like at the microscopic level and then also at the gross level, and what types of injuries you can see in different tissue types. First, we'll talk just very briefly about the various agents of cellular injury, meaning what types of things can physically damage a cell. And when we look at things in general, we have poisoning or toxicities, and we can also call those intoxications. There's physical damage we'll talk about, and there's also something called a deficiency. So when a cell is deficient in a primary nutrient, that's going to end up affecting the tissue and then the organ and then the organism. One example of a type of cellular deficiency would be a B12 deficiency, vitamin B12. And this is a specific type of nutrient that is taken in through various products. You can see some products that B12 is contained in. So these are dietary sources. And you'll notice you've got dairy, you've got eggs, you've got some poultry, and I think this is a lobster tail, chicken, cheese. So this should clue you into the fact that B12 is contained in animal products. It's a constituent of the phospholipid bilayered cell membrane, this plasma cell membrane. So anything that is ingested with a plasma cell membrane will eventually be broken down into component parts, and one of those constituents is B12. And B12 is so critical to a variety of aspects in the physiology of the body, one of which being the manufacturing of various blood cells. The stem cells absolutely need vitamin B12 and other things in order to become something like a mature red blood cell. So we would call a cell or an individual, we'll start there, an individual who is deficient in B12, that deficiency can present in two ways. The first being called a primary deficiency. And in that case, that would be a deficiency of the constituent in the diet. So an example of this would be a patient or an individual eating a vegan diet. And a vegan diet is one that includes 0 animal proteins. Anything without a plasma membrane does not contain B12. Plants and animals have different cell membranes. That means that individual in a primary way, because it is lacking in the diet, can become deficient in B12. There is also a secondary type of deficiency where there is adequate nutrient in the diet, but for whatever reason, there is an inability to process or absorb that nutrient. An example of a secondary deficiency of B12 specifically is called pernicious anemia. And pernicious anemia, first of all, emia is blood flow, and an means without. So I love that term. It's such a dramatic term, without blood flow. Pernicious anemia is a state of anemia, so a low level of hematocrit, a lower level of circulating red blood cells, which carry oxygen on them. And if you do not have adequate levels of circulating red blood cells, there's tiredness, there's pallor, there's going to be a lot of difficulties associated with this, and then an inadequacy or a low level of not only red blood cells, but also platelets and white blood cells. And the reason we see pernicious anemia, so the specific type of B12 deficiency, is not, again, because of a deficiency in the diet, but an inability to absorb vitamin B12. Vitamin B12 is attracted to a specific factor that's expressed by parietal cells in the gastric mucosa. So down in these gastric pits, we have cells that do a variety of things. And these parietal cells, they secrete something called intrinsic factor. And intrinsic factor binds to vitamin B12 so that it can go into the places of the body that can absorb and then utilize this B12. So if anything damages the gastric mucosa, one of the things that can cause this is gastritis, which is inflammation of the stomach. And you can see gastritis from chronic alcohol consumption and things like aspirin overuse. Those two substances, alcohol and aspirin, they physically can traverse or cross the stomach because of the way that it can travel through fat or water. So you get inflammation and then destruction of the cells along these gastric pits. So without intrinsic factor binding to B12, we can't utilize that B12. So there will be insufficient red blood cells. The production goes down because B12 is a constituent of the creation of these red blood cells. So you'll start to see symptoms of oxygen deficiency. So we start to see, as an example of this, something that's a little bit of a manifestation. So what I'm doing is drawing your attention to fluid underneath the meninges, directly above the cortex. This is a manifestation of this pernicious anemia. Way down the line, if this is not controlled, it can cause something called Wernicke-Korsakoff's syndrome. And this is a type of dementia that gives you memory loss in the same type of way that you would see in Alzheimer's type dementia, but it is a direct result of a B12 deficiency. B12 isn't something that can really be banked in the body. Any that could be saved really for short-term bases, really on the day scale, would be in the liver. And this is compounded by the fact that in chronic alcohol use, the liver is also damaged due to chronic overdrinking. Another term for Wernicke-Korsakoff syndrome is called wet brain syndrome. And that fluid here in your scan should lead you to the conclusion of why they call it that. There is an accumulation of fluid along the cortical structures, along with also a dilation and an increase in size of the ventricles where there is cerebrospinal fluid. So we see general cortical atrophy and then dilation of the ventricles. So we're getting actual destruction of cerebral tissue and then this resulting dementia-type disease. And this is all stemming, again, from this B12 deficiency. So when we say deficiency, it doesn't really mean like that's why you crave red meat or something of that nature. It's when you're deficient in a core constituent that's necessary to produce and keep in line, control is the word I was looking for, various activities in the body. So that's deficiency. Another type of agent of cellular injury is intoxication or poisoning, more commonly known. There are two types of ways that cells can become damaged in the poisoning sense. It can either be through endogenous, so things that are created within the body, or exogenous, things that are created outside of the body. Even though something is created in the body, that doesn't necessarily mean that we want it there, especially in high amounts. We can see endogenous types of cellular injury occur through poisoning, through things like impaired circulation. We might see metabolites, which are natural breakdown products of things like drugs or food, anything that's broken down by the liver into byproducts called metabolites. Metabolites need to be excreted at some point from the body, so if they do accumulate, that's going to alter the biochemical status of the body, leading to this intoxication state. Other things like CO2. CO2 is an acid. We know CO2 is, at the simplest level, our byproduct of oxygen utilization. We get CO2, which should be brought back up to the lungs and then exhaled. Because CO2 is an acid, if it is not removed from the body, we can see the pH of the body go down. And biochemical reactions don't occur outside of a very narrow window around 7.4 pH. So it's important that the circulation be intact to carry up the CO2 to the lungs, and also for the lungs to be active and healthy to remove the CO2. We can also see this in the case of bilirubin accumulation. Bilirubin is a byproduct of red blood cell breakdown, and it is a pigmented type of endogenous component. And when there is insufficient excretion due to low bile production or liver damage or some sort of obstruction in the bile pathway, we can see bilirubin build up in the circulation. And this yellow pigmentation can first start to color the nail beds and then the skin and the sclera, the white parts of the eye. And this is known as jaundice. So jaundice itself isn't a disease, but it is a sign that tells you something in the body is broken. Bile is not flowing the way that we need to, and the only way that we can remove bilirubin from the body is through bile, which will incorporate it into the feces for excretion. So these are just examples of ways endogenous products can build up in the body and cause intoxication. There's also a variety of genetic abnormalities where we can get the accumulation of waste products that would normally be removed, but because of the genetic code of this individual miscoding for proteins that are critical to this need, we see, again, byproducts that should be excreted or eliminated in some way retained in the body. We can see this in Huntington's disease, which is a neuronal disorder, causing choreatic movements and other types of motor manifestations. And in this case, we can see breakdown of nerve cells as a direct result of this disease. There's also PKU, another genetic abnormality where the individual is unable to break down the constituent phenylalanine, which then builds up in the physiology, altering the biochemistry and causing intoxication state. And then there's also exogenous cases of intoxication, which would literally mean anything from outside of the body brought in high quantities, causing a toxic state. So this can be examined in ways that you might be familiar with intoxication, so direct ingestion of substances or inhalation of substances, drugs of abuse, but also different gases like carbon monoxide can cause this. We also see this in an infection state where there is a colonization of the tissues by some sort of exogenous organism like bacteria, other microorganisms, fungi, or protozoa. So intoxication are high levels of substances causing misregulation of cells and then tissues and then, again, the entire organism. All right, and then we have traumatic injury. So this is when there is a physical force, some sort of external force damaging the cells in the body. And this flow chart is bringing you to trauma at the center and then giving you a variety of types of agents of traumatic injury. First, we have hyperthermia, so excessive therm is temperature. So excessive temperature, the body is too hot. This can happen due to things like electric current, touching a live wire, being, electrocuted, getting hit by lightning, solar radiation. When you get a tan, what you're seeing is melanocytes becoming damaged and dumping out some of the melanin, and that darkens the skin, but it's also a marker of injury. And we do know that too many of these suntans, especially burning types of injuries, cause a lot of damage over time, which we'll get to. And then there's also direct contact with things that cause hyperthermic reactions inside of cells, and that can be as simple as touching a stove or touching fire, something that's too hot and it injures those cells. And at the very basic level, this causes the unfolding of proteins, which are quaternary or tertiary in structure, and when they unfold, we call that denaturing. So they're denatured. And that's important because everything in the body, most things in the body function via shape. They need to be a correct shape in order to preserve their function. So when proteins become denatured, they lose their conformational shape, which is going to alter their function. So hyperthermia is a type of traumatic injury, and various things can cause hyperthermia in cells at the cellular level, which then damages the tissue. The opposite then of hyperthermia, we've got hypothermia, so low temperature. When cells are in contact with very low temperature for an extended period of time, at the cellular level, the cytoplasm, which is generally gelatinous in nature, not super watery, but a little bit more viscous. It does almost like ice cream. You get freezer burn on it. We see that in the cytoplasm. So we see ice crystals form in the cytoplasm after a while. And the cytoplasm is where things traverse themselves inside of cells. So up and down the cytoskeleton, we see proteins and vesicles move. We need to have that space intact for things to move through it. So that at the cellular level, becoming too cold causes cellular injury. Another type of injury is pressure injury, which is kind of just like what it sounds like. A pressure injury is pressure being placed upon a cell. And in the same way we can get a crushing injury to an organ or a tissue, like if I have a piano dropped on my hand, that's called a crushing injury, literally crushed. At the cellular level, when there's pressure on a cell, its cytoskeleton breaks down. So the same type of way, if, that piano fell on top of my hand, it breaks the bones. And then we don't have, the conformational shape and the function that we need. So when there is extended pressure upon cells, the cytoskeleton breaks down. It is an essential component of the cell for function. So that is something that causes traumatic injury and can definitely cause extreme amounts of damage to the tissue over time. Immune defects in general are referred to as an agent of traumatic injury. The immune system has a variety of functions, one of the most important being patrolling the body and then attacking, recognizing things that are foreign so that it doesn't cause a reaction in the body. All right, then we have ionizing radiation. We might know that radiation is not good for you. You should not be around it. You might have also heard that radiation is a way that we treat cancer. And the way that it's used in cancer therapeutics is to be targeted specifically towards tumors to cause DNA strand breakage. And that is where we're hoping to go with cancer treatment using radiation, targeting tumor cells or cancerous cells specifically, and breaking down their DNA so that those cells don't replicate. But in healthy tissues, we still see that same type of damage. We see DNA strand breakage and the development of things like reactive oxygen species, or what we call free radicals. forming, which alter the function of the cells. And then finally, I mentioned before that when there is an exogenous agent that enters the body, that's considered an intoxication or a poisoning. And an infection is when there is a full colonization of that tissue eventually, and we get a physical damage to that cell, which then may be damaged further by exudate development or fluid formed via inflammation. So this can also happen by the natural mechanisms meant to remove an infection. So just as I mentioned, exudate is a fluid formed via inflammation. When there's too much exudate, it can cause pressure to be put on surrounding cells and then breaks them down. Also, in an immune response, it can be very damaging to a tissue and cause its own injury. So those are all different types of traumatic injuries, which falls under our agents of injury in cellular. Next, let's talk about responses to those agents of injury. The body's job is to keep itself alive. The tissues want to keep the organs alive, and the organs want to keep the body alive. So the cells will adapt to lengthen survival the best that they can. And depending upon the length of irritant being in contact with the body, we can see various types of changes. The first one we'll talk about is hypertrophy. And hypertrophy, hyper is excessive, and trophy comes from trophic, which is going to refer to growth. So hypertrophy is excessive growth. So this is when the cells themselves become bigger. They need more mitochondria for more energy. And this can happen in things like skeletal muscle. So when you lift, you get swole. Those skeletal muscle cells, they get bigger because you're utilizing them more. They become hypertrophic. But this can also happen to other types of muscle too. So here's myocardial tissue. Here's normal myocardium. If anything stresses the system, high blood pressure, for example, or a blockage in the system, and the heart has to work harder. In the same way, you increase your reps or increase your weight. When you're working harder, those cells, they get bigger. So we see hypertrophied tissues, hypertrophied cells, bigger cells occurring in anything that has to work harder. So when this happens at the skeletal muscle level, we usually consider it cosmetic and great when there's hypertrophy. But when it does happen in internal visceral types of tissues, it can be very problematic. So we see hypertrophy in cardiac disease. We see hypertrophy of intestine smooth muscle in various types of chronic GI diseases. So hypertrophy is an adaptation to increased work, demands placed upon that tissue. And then the opposite then of hypertrophy is atrophy. A is without, so atrophy is without growth. In atrophy, instead of seeing cells getting bigger and these little guys inside, those are your mitochondria providing, energy. Those are your energy sources, really, of the body. If a tissue, for whatever reason, is not engaging in activity, the body will route blood flow and other means of energy sources, energy flow, away from that tissue. So the cells become smaller and the number of mitochondria go down, and we reduce the energy capacity of that tissue through those cells becoming adapted. So they become smaller and then engage in less energy, utilize less energy. There are a couple of different types of atrophy. The first one is referred to as disuse. So disuse atrophy, an example here would be the calf muscles, the shins, the lower leg muscles, essentially, of this individual. If there is a reason that prevents somebody from utilizing a part of the body, so the legs, the feet, the arms, those muscles are not being utilized, so therefore the body is going to route energy away from it, route blood flow away from it. And then the cells will then respond to that, again, by shrinking and reducing their mitochondrial expression. So less energy from less use. So that's disuse atrophy. The second that we'll talk about is called pressure atrophy. And pressure atrophy occurs not because the cells are, being underutilized and having, blood flow routed away from them. In this case, pressure atrophy occurs really through this kind of traumatic injury due to pressure. If you're not super familiar with the brain, this is a sagittal section. We've got our cortex out here, which is a little flat in this picture. It shouldn't look like that. And there's another thing that shouldn't really be there, and it's this big tumor right here. And when there are tumors in tissues, specifically malignant tumors, so cancerous tumors, they have a lot of mass to them. And when a tumor grows and it's thick, again, it has mass, it's heavy, what it will do is put pressure around or on the surrounding healthy cells, which, remember, they have inside of them their little cytoskeletons. And it needs its cytoskeleton to stay that way, to stay alive, to stay functional. So this pressure is exerted on these healthy cells, breaks down their cytoskeleton, and then they'll start to die because without their skeleton, they're non-functional. And now this tumor has another space to grow into. It can now encroach over to fill up that space where those healthy cells once were that it now crushed. That is one of the ways that tumors can grow. It's called expansion, and we talk about that later on in different chapters. But you can see that there was so much growth of this tumor that it caused a forward shift. So what you're seeing here in this flattened cortex is that it's made a lot of contact with the meninges and then the skull and has lost its nice normal round shaping. So pressure atrophy is pressure from something breaking down at the cellular level the cytoskeletal components of healthy cells, and then those cells dying, those cells becoming damaged. Okay, so we had hypertrophy. So remember, hypertrophy is big cells. That's different than hyperplasia, which is essentially more cells. Plasia comes from plasm, which for your purposes you can think about as substance. It's a Greek word that's referring to the term to mold, like you would mold a sculpture out of clay. So it's like a substance, a moldable substance. So excessive substance, hyperplasia. So rather than large cells, hyperplasia is more cells. So the cells themselves aren't getting bigger like you see in hypertrophy. Here's our normal cells. Here's hypertrophy. Cells are just bigger. In hyperplasia, whoopsies, I did something. These normal cells just start replicating. And that's essentially what it is at its very basic level. It is cells engaging in mitosis as a result of some sort of stimulus being released. One of the simplest things that you may have all experienced is hyperplasia of epithelial cells. When that happens, this was supposed to be an arrow, so now I'll draw an arrow. That looks great. It doesn't. I admit it. Let me just draw it over. Better. Epithelial cells, when there is an irritation, so this could happen when you first get braces and they initially tear up the inside of your cheeks and your lips, and then eventually it becomes a little thicker in there and it doesn't do that anymore and you can eat and smile without pain. This can also happen when you're wearing uncomfortable shoes and they rub up against your heel and it tears your foot apart that first day, but eventually it doesn't hurt so bad. Well, the reason for that was damage to those cells and that irritant signaling that we're going to have to protect that area. And it signals that there should be mitosis. And mitosis is when a parent cell, so if this is a parent cell, it splits into two identical daughter cells. They are identical to each other, and they're identical to the parent cell. So that's mitosis. Mitosis is generally okay. So hyperplasia, again, in epithelial cells, we see this with the development of a callus. This happens throughout the menstrual cycle. We see hyperplasia of the endometrium. So as we have changes to estrogen, if I draw something up here, let's pretend this is like an estrogen curve. As estrogen goes up, we start to see the thickening of the lining of the uterus, so that if an egg and when an egg is released at ovulation, and estrogen starts to go down, but progesterone starts to go up, there should be a nice thick lining here in the inside of the endometrium. If there is no thickened lining, the egg has nowhere to implant. If an egg then also does not implant inside of a uterus, then I just made this box into a uterus now. I'm committing to it. So if an egg does not implant in this thickened lining of the uterus, then it is expelled through the monthly menstrual cycle, M for menstrual cycle. And then the whole process starts over again the next month. So it's not bigger, intrauterine cells, it is more cells inside of the uterus. So this is something that we see, again, in an adaptation that occurs throughout the month in females, that occurs just due to irritation from braces, from baseball gloves, from bad shoes. So simple, just irritation causing more mitosis. We see this when you're sick and your lymph nodes get really swollen. There are B cells in lymph nodes, and we want to make more B cells and plasma cells in the case of infection and an immune response. So we have hyperplasia of those immune cells, which are precipitated in high amounts in the lymph nodes, so they feel larger, they get bigger. One cool thing that I love about the kidney is the kidney has a high amount of ability to become damaged and then replicate its various cells. So if somebody, for example, is born with only one kidney, sometimes they don't even know about it until there is a scan of the abdomen and it's noticed that there's only one kidney there instead of two. The reason for that is the kidney that remains, so the one that they were born with, undergoes hyperplasia until it is the size, basically, but mainly the functionality of two kidneys, so that the one kidney will take on the function of two, which is why you can see that happen with no symptoms. You also see that in the case that there's a kidney removed for whatever reason. The remaining kidney undergoes hyperplasia until the functionality of both kidneys are taken over. So again, this is an adaptive change. More growth, more cells to take on the function of tissue or to protect tissue that seems to be at risk. All right, so now is when we start to get a little bit closer to the danger zone. So hyperplasia is more cells due to an irritation. Now we're at metaplasia. Meta means change. And again, that plasia comes from that plasm, which is substance. So instead of, bigger cells or more cells, now we have different substance or different cells. So meta involves a change, kind of like the word metastasis. Stasis is location and meta is change. So metastasize, it moves, it's a change in location. So metaplasia is a change in a cell type. So again, this is due to an irritation. Hyperplasia is generally, it's still a longer type of irritation, like wearing shoes for multiple days at a time, braces that stay on the mouth for, weeks to months. Metaplasia can happen after much longer than that. So we see this after, decades. In this example I'm going to show you. So in the first scan here, you have normal tissue inside of the lungs. And these guys here, goblet cells, it's their job to make mucus. And wherever you see goblet cells, you should also see ciliated cells, because as mucus is produced, let me change my color to like green, even though not all mucus is green, but here's some mucus, and as that mucus is produced, the cilia should move that mucus so it can get out of the lungs. And that, my friends, is called clearance, the movement of mucus by cilia. So ciliated cells and goblet cells should go together so that there's good function, normal physiologic function. When people do things like smoke for a long time, so maybe they're at a pack or two a day, and they've been doing that now for 10, 15 years, what we start to see is metaplasia of the lungs. So this is what we would see inside of heavy smokers' lungs after extended smoking. What I want you to notice, first of all, is, where's the cilia? Well, it's not there, because in this type of metaplasia, what we see is an increase. So here we had our goblet cells, so we still have them here. So we see an increase in goblet cells, which means more mucus, right? So there's going to be more mucus. But now we have a loss of ciliated cells. So there's going to be an inability to engage in that clearance. All right, well then, who's going to move the mucus? Where is it going to go? How's it going to get down to the esophagus so it can be swallowed and digested? Well, the answer is you're going to have to do your own clearance. You're going to have to cough. The reason why we see this, I want you to notice how tough this tissue is. It's tougher, it's stratified, and the body has essentially changed to adapt to the fact that there's tar out here, there's nicotine, there's stuff coming in all of the time, and the body wants these tissues. So it has replaced now these nice ciliated cells with more goblet cells and tougher stratified tissue, but we've done that at expense of the cilia, meaning coughing is going to have to go up because there's no cilia to engage in that clearance. So again, this is a type of metaplasia, and this is in really, really heavy smoking after many years. So this should let you know that this type of damage, this is when we start to get into danger zone. This is adaptive, which means it can still also go back. This is a reversible change. If this person quit smoking, for example, this tissue would eventually, after many, many years, but it still happens, would go back to be expressed in this way. So metaplasia, we want to ideally reverse when possible. Because if it is not reversed, and now we have a different tissue type, which does not look like the parent tissue, then it becomes more likely that we make errors in copying those cells. The more protect, which is what we start to see in dysplasia. Dys means bad or deranged, bad or deranged. Dysplasia, a deranged substance. This is when there is very severe, prolonged type of irritation. And what differentiates dysplasia from metaplasia is it's still, these cells look different. They look abnormal. They're tough. They're stratified. But we start to lose the architecture. So we don't see cells, lined up, in layers the way that we should. It starts to lose that and it becomes more disorganized. We also see the prevalence of abnormally shaped, and we can see that through staining, nuclei of cells. When we have abnormally shaped nuclei, we call that pleomorphic. Pleomorphic cells have abnormal nuclei, and again, in mitosis, we have a cell, this is healthy, right? A healthy cell, when it splits, should form two daughter cells, identical to the parent cell and identical to each other. In dysplasia, these cells are looking less and less like the parent cell. So it's kind of like making copies of something, like maybe I made a copy from my book, and then you make a copy of it, and then you don't have enough money for a color copy, so you make it black and white, and then a page gets, bent. So there's a crease in it. And so every copy you make looks worse and worse. That's kind of like this process. We're not seeing this nice mitosis of, this cell looks exactly like this one, and it looks exactly like the parent one. Each time we're getting a splitting or replication of these cells, they're starting to look less and less and less like the parent cell. And this is when we're really in danger zone, because the more pleomorphic cells you have, likely you are to go to the next step after dysplasia, which is neoplasia, which is never good and not really adaptive. It is the result of dysplasia that was not resolved. Neo means new. We never want new tissue. That's not good. That's not an adaptive thing. Like, congrats on this tumor. Neoplasia is new tissue that doesn't resemble any other tissue that belongs in the body, and we don't like it. So because the next step is neoplasia, or cancer, more commonly referred to as cancer, we call dysplasia then pre-cancer. So you'll hear this term a lot, a pre-cancerous lesion. Speaking of precancerous lesions, this is a pap smear showing cells from the cervix. And what you see are cells that are stained to detect abnormal cells, abnormally shaped and sized, shape and size nuclei. And in blue, what we see are we've got some abnormal cells there. I want you to notice that in this pap smear, not all these cells are abnormal, right? Some of them are abnormal. Some look good. Some are normal epithelial cells. So this means, like in this case, we would get rid of all of this. Let's cut it out and let's remove that tissue. So here's the deal. Dysplasia is really the last step where you can get reversible damage, reversible change. If the irritant is removed, dysplasic cells, because cells are always replacing, they're always growing, we can still see tissues return to normal. But once we get to neoplasia, we can no longer reverse that change. Cancerous cells, so full-on pleomorphic cells, which eventually make up a tumor, can't go back to looking like normal cells. All right, so as I mentioned, we can adapt so far, and then once we get to a certain point, there's no more adaptation. And we mentioned that dysplasia can eventually become neoplasia. And if we had to kind of put neoplasia or cancer to the simplest form, we would say it is when a cell loses its capability to remove itself from the pool. It loses its ability to engage in a normal, controlled death response called apoptosis. All right, so let's talk about what that is. So when we have damage to the nuclei, again, like we said in a pleomorphic cell, those are abnormally shaped and abnormally stained nuclei. We need that cell to die. We don't want a cell with an abnormal nucleus making copies of itself, which are going to look less and less like the parent cell, just hanging out in the body, precipitating in tissues and making up tissues, because now they're going to take over and do what they want. So ideally, when a nucleus becomes damaged, when it doesn't look right, when it doesn't look normal, it should, as a cell, kill itself or put out a message that it's time to die so that it can be removed from the pool of cells in the body that are able to be copied. Different types of damage to the nuclei, just in general, this in A, this is our nice normal cell, happy. In B, we have what we call karyolysis. Lysis is to break apart. This is very lightly stained nuclear material that's very sparse in the cell, so it's like an explosion, almost left little ashes of the nucleus around the cell. We have pycnosis. Osis means development of. Pycnosis is when we have condensed nuclear material, so it's imploded essentially on itself rather than looking like a nucleus with its nuclear membrane, a nucleolus. It doesn't look like that. And then finally, we have karyarexis, which is what we call a fragmented nucleus. That's what you see here. These are fragments which means that it's kind of together, kind of like a book of stamps, but it's really easy to flake those pieces apart. So again, when we see this damage to the nuclei, we don't want that cell copying itself. That is an irreversible change. We can't just reverse this and make those go back to normal looking nuclei. That cell die. And programmed cell death to remove those types of abnormal cells with crazy looking nuclei from the pool is called apoptosis. Sometimes people don't say this P and they say apoptosis. You can say it however you want as long as you say it with confidence. I say apoptosis because that's what my professors taught me when I went to school, and that's what I'm comfortable doing, and I don't want them to hear me say it differently. So sorry, it's apoptosis, professors. All right, so in A, we have nice, normal, healthy tissue, right? Yay! Then we have a cell that has become pleomorphic. Some sort of irritant was introduced into the body, and this cell adapted, and eventually we got a pleomorphic cell. It's got an abnormal nuclei, so we hate it. Thanks, I hate it. So what that cell does is put up a little signal, and because it's hard for a cell to kill itself, but it can put up a little flag and say, hi, I need to die. I don't want to, ruin my healthy brothers and sisters in this tissue by taking the chance that I'll be copied, because then I'm going to take over this tissue and I'm going to look crazy over here as, my daughter cells will take over. So I need to die. So this awesome enzyme comes by. It's got a cool name. It's called, it's a caspase, but it's also called the executioner enzyme. And it's like, thanks for calling me, time to die. So it comes over and it releases stuff and it kills that cell, and any fragments left over by that cell are just engulfed by the other cells that once loved it and considered it family. Now they've took it in for their own nourishment and filled in the place that cell was. And now look how happy this tissue is. We remove the pleomorphic cell. There's no abnormal looking cells in the area because of apoptosis. Now this tissue is healthy and we don't have to worry about it. Yay, we love apoptosis. And you've all experienced it before, maybe, at least this one. We all experience apoptosis in our synapses. Synapses are the space between neurons. And fun fact, intelligence has not been correlated with the amount of neurons that you have. However, it has been correlated with the amount of synapses you have, so the amount of connections you have. And when all of us are born, we kind of look like this. We take a cortical slice. There's a couple neurons there. They're doing basic things. But if you've ever been around like a newborn, they don't do a lot. You can put them somewhere and then leave them like for 3 hours and they can't move. It's great. But what we see is that through interacting with their environment, they start to build up these synaptic connections, which by about the age of 7, it's crazy. If you've ever been around a 7-year-old, you can kind of see that their brain looks exactly like this at the cortical level. They make connections to everything. There's way too many connections here. So around the age of 15, we start to see these things being altered and kind of cut back. And so it's a gardening term that they use. It's called pruning. So synaptic pruning. And pruning would refer to like when you're taking care of your plants. Let me try to draw a plant right here. Yeah, it looks good. So when you're taking care of your plants, when something starts to overgrow, you cut off that piece. And now it doesn't exist anymore, and it reroutes the energy to the other parts of the plant, and they grow and they become healthy. So you can, have the healthy parts grow and thrive. So we see the synaptic pruning, happen again around adolescence, and this occurs until, forever, until about the age of 24, when the cortex becomes what we consider adult. But again, this is controlled cell death. So that's an example of apoptosis happening. I said it, sorry, it's apoptosis. So those cells die because we don't want them. That's controlled and it's healthy. We also see this after nursing. So this is actually from a mouse. And what you're looking at here is good way to describe this simply. So you're looking at mammary glands, and the cells that secrete milk are called mammotrophic cells. So here we see all these mammotrophic cells. And in this study, there were mice that were lactating, so they had just had a litter and they're making milk. And this is a normal group here, control group, who aren't given any type of drug. And in this study, this other group was given a drug. And what that drug did was block prolactin, which is the hormone that causes milk to be made. And when prolactin is around, so when PRL is around, not only do you get milk, you also get hypertrophy and hyperplasia of these mammotrophic cells. When lactation is done, when weaning occurs, or in this case, when this drug was given to block lactation, we see that these cells decrease in number and get smaller. This is controlled apoptosis. We don't have active mammotrophic cells making milk forever, thank goodness. They send out a message, the caspase, that executioner caspase comes around, it kills them, and then the tissue goes back to normal. That says nothing about the elasticity of the skin around it, but at least the tissue does. All right, so that's apoptosis, which is controlled cell death. There's also non-controlled cell death, and that's not called apoptosis, that's just called necrosis. Osis is the development of, and necro is death, the development of death. When a cell becomes damaged due to one of these agents of injury we talked about, so maybe it's poisoning, maybe it's electric current, we see necrosis of cells. So when a cell is damaged to a point where it's going to die, where it needs to die, it, first of all, releases various products, like it doesn't mean to, but its contents are released out into the body, which then targets phagocytic cells and enzymes to that area to start eating up the remnants of that cell. And also, by its own accord, these little holes here are showing you the places where the lysosome, so the stomach of those cells, were unzipped. And what that does is let out the acid inside of the lysosome, which pokes little holes in the membrane. So then the more holes you have in the membrane, the more contents leak out. And the more contents leak out, the more enzymes and phagocytic cells come to the area. So that is necrosis. Necrosis is not reversible. Once death occurs, it is there. Now let's talk about what that looks like. Then you might know that.
All right, so in pathology, you're going to be looking at dead tissue, right? So in cellular necrosis, dead tissue looks like dead tissue. How are you going to know when it died, right? It's all going to look really similar. And so the answer is almost always, like for everything in pathology, put it underneath a microscope and see what it looks like. And in really near death, the tissue, the first type of presentation that you'll see is called coagulation necrosis. So this is a very recent type of tissue death, immediately following necrosis of the tissues. The tissues will appear firm, their architecture will be maintained. When you do some sampling at that area, some proteomics, you'll see high levels of acid, calcium, denatured proteins, and when you look at the cells, you'll see that there's a loss of nucleus. So in this picture, you're seeing a type of burn. Again, this would be like, well, how did that happen? When did it happen? So the answer is looking at the tissues. And if it happened most recently and it shows those characteristics that are just mentioned and are written underneath the picture, we call that coagulation necrosis. Here we see something similar, but it is in usually neural tissue. We see this type of necrosis, liquefactive or liquefaction necrosis. It is liquefactive in nature. So this, again, this is an immediate type of response that you would see with tissue death. And this is generally inside of some sort of encased organ. Here you see some sort of damage due to an infarct. And when we have that type of damage, this is not a hemorrhagic infarct because we can see that there's white tissue. We also know that there was some sort of damage and swelling because we see this shift in the brain. So we call this a midline shift. This is the midline of the brain, and the ventricles should be on either side of it, kind of like a butterfly. But we see that they've shifted over this way, where we see the relatively normal cortical tissue with our gray matter and our white matter. But over on this side, we can see that there was some pronounced tissue death. Again, we see this white infarcted area. We see this area of liquid. That's why we call it liquefactive in nature. You will see this type of necrosis, again, normally in neural tissue. The brain is one of the most common places to see this. And this is telling you that this was a recent time of death of these cells. And these are hydrolytic enzymes. Lytic is from lysis. So, and hydro is water. So hydrolytic enzymes are going to be key in the breaking down of these cells. So liquefactive necrosis is kind of like coagulation necrosis, but we see it in places like neural tissue where things are encased inside of the body. So tissues appear wet and they are dead. Again, this is irreversible. We can't go back from here. There is caseous necrosis. So remember, coagulation necrosis is near immediate death of cells and then the tissue. So the tissue is firm, architecture is maintained. When necrosis goes on for longer, we'll start to see changes in the tissue. Again, both we're going to see this at the cellular level inside of the cells, and then also at the gross level. So this is a type of necrosis we can see at the gross level. Caseous comes from the word casein, which means cheese. So gross, right? So this is caseous necrosis, and these are little areas of caseous necrosis. So when something caseates, it takes on this cheesy type of appearance. So these are dead cells, and they've started to caseate. And caseous necrosis is a combination of coagulative, so that immediate tissue death, but also liquefactive in that there's hydrolytic enzymes here breaking down the cells, and we can see the shininess, this wetness of the tissue. And so again, this lets you know that we have, relatively a recent death, but definitely longer than coagulation necrosis. When you smell caseated tissue, you'll never forget the smell, I promise you. So caseous tissue does have a very specific scent to it. So let's say you were changing a bandage, for example, and something smells bad and you start to see this white tissue. So you can make the judgment that this had been there for a while, for definitely a couple of days, and this has started to caseate. So it's not like coagulation necrosis where things are dry. It's now started to caseate. It appears a little bit slippery, a little wet, and it's got that cheesy white-like appearance. We have fibrinoid necrosis. So oid means kind of like. So this is cell death, but in this case, we're not seeing really fibrin. We're seeing these weird little complexes, which are described as fibrinoid. And what you're looking at here, this is actually from the kidneys. And this is showing you that these cells have died and their contents are leaking out. And what is in these little fibrinoid complexes are a mix of immune complexes, which precipitate and form when there's a high amount of antibody to antigen ratio. So there's too much antibody for the amount of antigen. And rather than getting these really large accumulations of antigen antibody moving throughout the bloodstream and then being neutralized by the spleen and eventually excreted, we see that these little immune complexes can get stuck in the microcirculature, like in the glomerular vessels of the kidneys, which is what you see here. So the fibrin is leaked, and then we see these immune complexes associated with it. So fibrinoid necrosis is accompanied by these little fibrinoid pathologic markers. So that tells you that fibrin has leaked, and there are these immune complexes, which is a type 3 hypersensitivity. So that lets you know that this is immune, specifically a hypersensitivity mediated reaction in its basis. All right, now we've got the fun ones. We've got gangrenous necrosis. So death, the development of death with gangrene. So when tissues are dead for a specific amount of time, again, you can't tell, they all look pretty similar. So you have to look for things that make them different, right? How are you going to know how long it's been a dead tissue for? Well, you look for different things. We know if we see this white cheesiness, which we can actually see here, so that's some caseating tissue. It doesn't look like coagulation necrosis. So we see some caseating tissue, some caseous necrosis, but the predominant finding, I would say, that's abnormal is this black tissue. So if you can look at what part of the body this is a mustache, so this is an upper lip, we've got lower lip. So there was some sort of cellular death, which then progressed into tissue death. And what we see here, because this is exposed to air, we will have the deposition of bacteria. And when there is bacteria that is living in an area that is inside of the body, it will colonize it. So when tissue appears black, it's usually because of an oxidation reaction. So dry gangrene happens in coagulative necrosis, where you see this black appearance of tissues because of the oxidation. So in order to have dry gangrene, you have to have oxygen around. So the tissues have to be in some ways exposed to air, because any tissue that becomes ischemic will attract anaerobic bacteria to it. That can be on the inside of a thrombus, it can be in a heart valve, it can be anywhere. But when you have eventually oxygen reaching it, you get the oxidation of that tissues, and then it turns black. We can also see a mix here of liquefactive necrosis. Remember, that happens inside of the body. And that can cause what we call the presentation. Just saw here, this is dry gangrene. It's literally dry. It's on the outside of the body. Tissues are oxidized. They're black and dry in appearance. Of wet gangrene. So wet gangrene is when there is no oxidation, so we don't see black here. We see a red, open, weeping sore. This tissue is all dead. It's still definitely dead. And the term gangrene means that it's been colonized by bacteria. Dry means that it's outside, exposed to air, and oxidized. Wet means that not only is it a wet appearing wound, it means that there is no exposure to oxygen. So this is on the outside of the foot, it looks like, right? So how would we get wet gangrene on the outside of the foot? Well, this is something that you'll see when there are constrictive things on the feet. So we see a lot of this, and there was a history of this in like World War I and II, where soldiers had their boots on for days or maybe weeks at a time, and their feet were wet, and we're not getting any oxygen down there. If there was damage to the tissue and it starts to die and then bacteria gain entry to it, we'll start to see these types of eating away at the tissue. But because there's no oxygen to oxidize this, we just see this open, gaping wound. Okay, so that's just the term. So we've got necrosis in general is tissue death. We've got coagulation necrosis, which is firm, it just died recently. We've got caseating necrosis, which is coagulative, gone on for a little bit longer and a little bit more colonization of the tissues with precipitating enzymes. And then we have necrosis or necrotic tissue that's been colonized by bacteria, and we call that gangrene. And again, that can either be dry gangrene or wet gangrene. Here we see it. Okay, final marker of cellular injury is calcification. So when you're looking at tissues, and again, you want to know how long ago that tissue died, that tissue became injured due to cellular injury at the very low level of the cell. In certain tissues, like this is a heart, this is a cross-section of a heart, we see its septum here. When there is prolonged damage to a tissue and there are high amounts of calcium in the fluid around it, when a cell becomes damaged, the calcium can precipitate in those tissues, and calcium forms crystals. So we get, after long term, the deposition of calcium crystals, and we call that calcification. So calcium deposits after the site of necrosis, that lets you know generally, if there is some sort of calcification, this injury is very long-standing. You won't see calcification of a really recent damage in the heart. You'll see calcification of long-standing damage in the heart and dead cells that have been there for a long time. So those are the different gross appearances of necrosis. All right, so quickly, exam questions. All of the following are potentially reversible. Okay, let's see. Necrosis, is that reversible? No way. Once you're dead, you're dead. Metaplasia, I'm pretty sure we said that can go back after a while. Atrophy, well, if I broke my ankle and my calf atrophied and now I can walk again and it's the same size, so yeah, that's reversible. Hyperplasia, can I get rid of a callus? Let's see. Yes, I absolutely can. So our one that is not potentially reversible is necrosis. Death is death. We cannot go back. All right, increase in cardiac size and function. So bigger means more function, more capability of function due to increased workload. This is pretty easy. Is it atrophy? Nah. Is it hypertrophy? So it wasn't lack of growth. Is it extensive or excessive growth? That sounds pretty good. Is it functional? So a functional increase in size? I don't think we said anything about that in this lecture. No. If the heart gets bigger and it's functioning more, is it inflamed? No, that doesn't sound right. So hypertrophy is a good answer, and it is the correct answer. All right, we're done. Stay smart.